Good morning everyone. Welcome to today's lecture. Today we are going to learn and understand about the transcription process in prokaryotes. And we will try to learn and understand this topic under the subheadings. So under the introduction we are going to discuss some important compounds, important concepts, and we are going to learn about the RNA polymerase, the structure and how it organizes. It assembles over the DNA polymerase. And under the mechanism of transcription, we will discuss the clear mechanism of RNA synthesis, means how the RNA is synthesized from the DNA molecules. So this includes 3 steps like mechanism, initiation, elongation. And lastly, we will see what are the inhibitors of the transcription process. And we will start with the introduction process. So under the introduction, we will discuss about the central dogma of life or central dogma of biology. So this is very, very important concept to understand. So here, so this diagram is showing that, so the DNA will produce a single copy of DNA. If the DNA is able to synthesize DNA, and that particular process is called replication. Means here the 2 DNA strands will separate and synthesize their own complementary copies. That particular process is called replication. And the replication occurs to produce its own copy so that this particular DNA molecules can be transferred to the daughter cells of the daughter cells. And next process is this particular DNA. If the DNA involves in the synthesis of RNA molecule, and that particular process is called transcription. So, and then if the RNA involves in the protein synthesis, that particular process is called translation. This is how the information which is there in the DNA molecule is transferred into the cells, and the cells express different kinds of proteins. So during the replication process, there is a similarity, means replication and transcription process are somewhat similar in nature. Why? Because during the, replication and transcription, the polynucleotide synthesis occurs. So this particular replication process is carried out by the enzyme DNA polymerase. During the replication process, so there are, in prokaryotes, there are 3 types of DNA polymerases: DNA polymerase 1, polymerase 2 and polymerase 3. Among these 3 polymerases, DNA polymerase 3 is the important enzyme which involves in the polymerization or synthesis of daughter strands to the parent strands. And here, if this particular DNA molecules involves in the synthesis of RNA molecule, and that particular process is called transcription. So during transcription, so the RNA molecule is synthesized, and this is this can be considered as first level of gene expression. So here in the first step, the DNA is producing its own copies, and in the next step, the DNA is able to express itself, means the DNA is coding for the RNA, and this is considered as first level of gene expression. And then this particular RNA, which is produced through the transcription process, involves in the synthesis of the protein molecules, involves in the synthesis of different kinds of proteins through the process called translation. And this is how the genes this is how the information which is there in the DNA molecule is decoded to the proteins. So this is an important concept, and which is called central dogma of life or central dogma of biology, and most of the cells, the genes will express in this fashion only. And now we will move forward. So here we are going to learn about the transcription process. First we will see what is meant by transcription. So transcription is nothing but the process of synthesis of RNA from the DNA by the DNA dependent RNA polymerase. So here transcription, through the transcription process, the RNA molecules are synthesized. So the synthesized RNA molecules may be of coding RNAs or non-coding RNAs. Coding RNAs means this particular RNA molecules further involves in synthesis of protein molecules. So that's why these RNA molecules are called coding RNAs. And one of the coding RNA is mRNA or messenger RNA. So this messenger RNA involves in the synthesis of the protein molecules. And non-coding RNAs means this particular RNAs won't involve in the synthesis of any other molecules. So the most important non-coding RNAs are rRNA, tRNA. And not only these two RNAs, there are some other non-coding RNAs also do exist, like guide RNA, microRNA, and so on. So those particular non-coding RNAs are not necessary to study under this particular topics. So here if you can understand what is meant by coding RNA and what are the non-coding RNAs, that is enough to understand this particular topic. So here this particular coding RNA, the messenger RNA which is produced during the transcription process, will have a message which is received from the DNA molecule, and this particular message will be decoded by the ribosomes to produce the proteins. And then this particular non-coding RNAs, the important non-coding RNAs like ribosomal RNAs and tRNAs, also involves in the process of protein synthesis. This particular ribosomes are very essential. This particular rRNA are the components of the ribosomes, and the ribosomes involves in the protein synthesis process. The ribosomes are called protein synthesis factories, where the protein synthesis occurs. So the ribosomes can be called as ribozymes. And this particular tRNAs, which involves in the transfer of amino acids from the amino acid pool to the site of protein synthesis. Where is the site of protein synthesis? On the mRNA and in the ribosomes. So this particular tRNA involves in the transfer of amino acids from the amino acid pool to the site of protein synthesis. So these are different types of RNAs synthesized through the process of transcription. And next, so during the central dogma of life, we have learned that first the replication occurs. Through the replication process, the DNA replicates to produce its own copies, so that the information which is there in that DNA molecule can be passed on to the other daughter molecules. And during the transcription process, the RNA molecules will be synthesized with the help of RNA polymerase. And these two processes are mechanically similar. But there are certain differences between the replication and transcription process. So what are those differences? So the replication will be carried out by the enzyme DNA polymerases. We already mentioned that this particular replication process will occur with the help of DNA polymerase, and in specific DNA polymerase 3. And the remaining DNA polymerases like DNA polymerase 1 and DNA polymerase 2 involves in the repair mechanism and proofreading activities. But the DNA polymerase 3 involves in the process of replication. This particular DNA polymerase 3, the single DNA polymerase 3 will replicate the two strands of the DNA molecule. So that is one difference, means in the replication DNA polymerase involves in the replication process, and in the transcription RNA polymerase. So here the enzyme involved in the transcription process is RNA polymerase. And during the replication, the entire genome of the species will undergo the replication process, so that the entire information which is there in the parent cell can be transferred to the daughter cell. But if you see the first level of gene expression, that is nothing but during the transcription process. So the selected regions of the genome will undergo the transcription process. So the entire genome won't undergo the transcription process. So the selected regions, the given region of that particular genome will undergo the transcription process. And that particular so the number of copies produced may be high. That depends on the requirement of the cell. So this transcription process means the high rate of transcription process. If the cell requires high amount of some particular protein, then that particular region will undergo transcription more number of times. So this particular selectivity is also another difference. So means during replication process, the entire genome of the species will undergo replication so that the intact genetic information can be passed on to the next generation. But during the transcription process, only selected regions of the gene will undergo the transcription process. And next one is proofreading activity. So during replication process, the replication process is very accurate process. So it will check that there is no a single base pair mismatch. So without any mismatches or without any single mistake, the DNA will undergo the replication process. So comparatively, the DNA replication is very accurate process when compared to the transcription process. And these are some differences between the replication and transcription process. So once we will see what we learned first is, we try to understand the we learned the concept of the central dogma of the life. So what is mean by central dogma of life or central dogma of biology? So which is nothing but if the DNA is able to produce DNA copies, means that particular mechanism is called replication, and which is carried out by the DNA polymerase III in case of the prokaryotes and other DNA polymerases in case of the eukaryotes. And from the DNA will show expression. So if the DNA involves in the synthesis of RNA molecule, that particular process is called transcription, and that can be considered as first level of gene expression. And then we learned about the differences between the replication and transcription process, and further we will move forward to understand what exactly transcription process involves. And before entering into the actual mechanism of transcription process, first we have to learn and we have to understand about the RNA polymerase. So here it is mentioned that in prokaryotes only single RNA polymerase involves in the synthesis of all kinds of RNAs. So this means that one RNA polymerase enzyme, the single RNA polymerase enzyme will involve in the synthesis of rRNAs, tRNAs and mRNAs. So all these kinds of RNAs which involved in the protein synthesis will be synthesized by the single RNA polymerase enzyme. And this particular transcription process is called de novo process. De novo process means here to initiate this particular transcription process, so the RNA polymerase won't require any primers. So here there is no necessity of the RNA primers or some primers to initiate the mechanism process. So that's why this particular transcription process is a de novo process. So here it directly initiate the polynucleotide chain synthesis. And next one. So this how this RNA polymerase will assemble. So here this particular RNA polymerase is composed of subunits 2 alpha, beta, beta prime and sigma subunits. So here you can see the structure of RNA polymerase. So it is composed of 2 alpha subunit, 1 beta and beta prime subunits. So this RNA polymerase exists as a core enzyme and holo enzyme. So the core enzyme is nothing but the RNA polymerase with 4 subunits like 2 alpha subunits, beta, beta prime is called core RNA polymerase enzyme. And if this particular core RNA polymerase enzyme is binds with the sigma subunit, then this particular enzyme is called holo enzyme. The holo enzyme of RNA polymerase can only initiate the process of transcription. But before actual initiation of the transcription process, this core subunits of the RNA polymerase can also start the process of transcription. So first, this core enzyme of the RNA polymerase, the core enzyme is nothing but which is having 4 subunits like 2 alpha, 1 beta, beta prime. So these 4 subunits constitute the core RNA polymerase, and this particular core RNA polymerase will go and bind on the DNA polymerase and can start the process of transcription, but that particular transcription process is not a strong or not a successive one. So why? Because it is without an important sigma factor, so it cannot initiate the proper transcription, but it do transcribe some few base pairs, but that will results into the abortive transcription process. So here an important thing to understand is the transcription in prokaryotes is called coupled transcription. So why we are mentioning this means, so why it is called coupled transcription means, in prokaryotes there is no clear demarcation between the nucleus and cytoplasm. The nucleus or the genetic material which is there will freely distributed in the cell, and there is no clear nucleus in the prokaryotes. So during the transcription means, here in prokaryotes the two processes occur simultaneously. So means before the completion of the transcription, the translation process occurs. So here you can see, so here this particular transcription process is still on, means the transcription is not yet over. But here the ribosomes have started reading the message which is there in this particular mRNA to produce the proteins. So means here the two processes are carried out simultaneously. Transcription and translation are occurring simultaneously. So that's why this particular transcription process in prokaryotes is called coupled transcription. If you see, if you observe in eukaryotes, the transcription is separated, means the transcription will occur in the nucleus, and from there the synthesized mRNA molecule has to reach the cytoplasm after post-transcriptional modifications. So it is locally separated. So in case of the eukaryotes, the transcription process will occur in the nucleus, and after the synthesis of mRNA molecules, the synthesized molecule is called transcript or primary mRNA, and this particular primary mRNA will undergo some modifications. After the modifications, that particular mRNA enters into the cytoplasm, and in the cytoplasm the protein synthesis will occur. So that happens only in eukaryotes, but in prokaryotes the process of transcription and translation occur simultaneously. So that's why it is called coupled transcription. So now we will try to understand the actual mechanism of transcription. So what it involves, and how the initiation occurs, and how the elongation occurs, and how the termination occurs. So the mechanism of transcription can be understood under the 3 subheadings: initiation, elongation, termination. So initiation is nothing but the starting point where the transcription will start, and elongation will be continued up to some regions of the DNA, and then it will terminate at a certain specific sites. So first we will see, before starting the initiation process, first we will try to see the promoter sequence. So the region where the transcription actually starts is called starting point of transcription, and it can be represented with the +1, and the region which is present towards the left side of that particular starting point is called upstream region, and the region which is there towards the right side of the starting point is called downstream region. And here if you see the promoter region, so this particular promoter region will be recognized by the RNA polymerase, and this RNA polymerase will come and bind to this particular promoter sequence. Then only the process of transcription will start. So if you see the promoter sequence, so this is all, so this region, all this region will come under the promoter sequence. So here the important regions to study, important regions to remember are these two. So -10 box, so which is here the sequence conserved in this particular region is TATAAT. So this particular region is also called Tata box, and this particular region is called -35 region have a consensus sequence like TTGA CA. So the sequence in these two particular regions is highly conserved, and this particular two regions are called core promoters. Means without this core promoter, the RNA polymerase cannot bind over the DNA and cannot start the process of transcription. So this particular core promoter region is very essential for the binding of the RNA polymerase and to start the process of transcription. So along with the core promoters, there are some up elements. Up elements is nothing but upstream promoter elements. So this particular upstream promoter elements were first recognized by Richard scientist in the on the DNA, which is coding for the ribosomal RNAs. So this particular up elements and enhancers will enhance the rate of transcription process, means will increase the transcription, will increase the rate of transcription. So the enhancers and up elements increase the rate of transcription, and these two regions like Tata box, which is also called Pribnow box, which is named after the scientist David Pribnow, who identified this particular -10 region in the promoter sequence. So the -10 box and -35 box are very, very important regions in the promoter. So this particular these regions will be recognized by the RNA polymerase. So this is the promoter sequence in case of the prokaryotes. So this majority of the prokaryotes will have these two regions, these -10 and -35 consensus sequences. So this is the promoter sequence. So the initiation, how the initiation occurs and how the RNA polymerase will recognize this particular promoter sequence, and how actually the initiation, means the unwinding of the double stranded DNA occurs, and how the initiation will occur. So the RNA transcript is nothing but the RNA molecule which is synthesized during the transcription process is called nascent RNA or primary transcript. And then transcription unit. Transcription unit is nothing but, so here a specific region which is having three components like a promoter and a RNA coding region and a terminator sequence are called transcription unit. So this particular transcription unit is composed of is essentially have the three elements. One is promoter, so where this particular promoter is recognized by the RNA polymerase and will start the process of transcription, and then a region, so which will encode the RNA molecule, and then a termination sequence, so which will stop the process of transcription process. So that is called transcription unit. And this particular transcription unit you can consider as synonymous term of the cistron. Cistron is nothing but the coding region of the DNA, or the region which is expressing the DNA which is expressing is also called cistron. So the transcription unit may be a synonym of the cistron. And then in the initiation process, so first the closed promoter complex will form, and that particular closed promoter complex can be converted into open promoter complex. So this particular conversion process is very, very important. So if it forms open promoter complex, then we can conclude that the initiation process has occurred. and this closed promoter complex is nothing but the RNA polymerase with its four RNA polymerase, core RNA polymerase will bind over the DNA polymerase at the promoter region, and it looks like a closed promoter complex. And then sigma factor, the fifth subunit of the RNA polymerase, the sigma factor will come and direct this particular core enzyme to the specific regions like -10 and -35, and after the binding of this particular sigma factor, the closed promoter complex will convert into open promoter complex because of the some conformational changes in the RNA polymerase enzyme. And this is an important step in the initiation process, and this we can conclude that the initiation process has started. And the unwinding region may be 10 to 17 base pairs can be unwind for the transcription process to occur. So this open promoter complex, which is formed after the binding of the sigma factor, is called a transcription bubble. So in the transcription bubble, you can see the two DNA fragments, the two DNA strands are separated. So this is one DNA strand and this is another DNA strand. So the strand which is among the two DNA strands, only one strand will code for the RNA molecule. So the strand which is coding for RNA molecules is called antisense strand or template DNA strand. So here in this diagram you can see this particular lower strand is coding for the RNA molecule synthesis. So you can consider this particular strand as antisense strand or template strand. So this particular template strand involves in the RNA synthesis process, and the other strand is called sense strand, which is not involving in the transcription process. You can consider this as another difference between the replication and transcription process. So here this particular transcription bubble will move further over the DNA fragment to synthesize or to continuously synthesize a specific gene into the RNA molecule. So what are the conformational changes? How exactly this particular sigma factor will assist the RNA polymerase to initiate the transcription process? So first also we mentioned that this particular core polymerase without sigma factor can also synthesize the RNA molecule, but it can synthesize only few RNA molecules. So it cannot initiate or it cannot synthesize a lengthy RNA molecules, and it results into the abortive transcription process. But after binding this particular sigma factor alone can drive this particular core RNA polymerase to the -10 and -35 regions. So after binding to these two regions, then the conformational changes occurs in the RNA polymerase, so that this particular RNA polymerase can hold on to the DNA molecule and can start the RNA synthesis process. So this particular sigma factor is very important one in the transcription factor, or to understand the RNA polymerase. This particular sigma factor is divided into is composed of 4 regions: region 1, region 2 and region 3, region 4. So the DNA which is present between the region 1 and 2, the alpha factor which is there between region 1 and 2 will specifically loosens the non-specific interactions between the RNA polymerase and DNA. So this particular sigma factor can be divided into 4 regions. Each region is having its own specific function: region 1, region 2, region 3, region 4. So this particular sigma factor is divided into 4 regions. So the region between the 1 and 2 helps to loosen the non-specific interactions of the RNA polymerase on the DNA molecule. And then the region 2 is subdivided into 2.1 and 2.4. So this particular 2.1, the region 2.1 of the alpha factor will help to bind, will help in the formation of the holo enzyme, and this particular region 2.4 of the holo enzyme of the sigma factor will assist in the recognition of the -10 box, which is nothing but Tata box. So the region 2.4 of the sigma factor will assist or will help in the binding of this particular sigma factor to the -10 region, and then region 4 is also subdivided, and 4.2, the region 4.2 of the sigma factor will assist the RNA polymerase to bind onto the -35 regions. So this is how, so this is the specificity of the sigma factor which assists the core enzyme or core RNA polymerase to bind onto the promoter sequence, so that the process of initiation can start. So after binding exactly onto the promoter sequence, onto the consensus sequences, the RNA polymerase will undergo some conformational changes, so that the transcription bubble means so that the RNA polymerase can unwind the 2 strands of the DNA. So here you can see the transcription bubble. Here the 2 strands of the DNA are in a melted condition, means the 2 strands are in an unwind condition, so that 1 strand is available for the RNA polymerase to start off the process of transcription. So the process of transcription occurs in 5' to 3' direction. So always in the why it is in 5' to 3' direction means, so always in the 3' direction there will be a free hydroxyl group, and to the coming nucleotide triphosphates will bind to this particular 3' hydroxyl group. So this is how initiation occurs. So once we will see what we learned in the mechanism of the transcription. So under the mechanism of transcription, so we will learn about the initiation, elongation and termination process. So before understanding the initiation process, we have to have an idea about the RNA polymerase and what are its components. So the RNA polymerase is composed of 5 subunits. So if an RNA polymerase with 4 subunits is called core enzyme. Core enzyme means which is having only 4 subunits. What are they? What are the 4 subunits of the enzyme? 2 alpha subunits, beta, beta prime. So these 4 subunits constitute the core enzyme of the RNA polymerase. So this particular core enzyme can also start the process of transcription, but which is not an accurate or which is not a successful transcription. So this particular core polymerase can synthesize only few base pairs, and after that the transcription process will halt. So like this, the core RNA polymerase will slide over the DNA polymerase until it is bind with the sigma factor. So then who is leading this particular RNA polymerase means it is sigma factor. So this particular sigma factor is very, very essential for the proper transcription process to start. So this particular sigma factor will come and bind to the core RNA polymerase enzyme, and this particular binding of sigma factor will helps the RNA polymerase to recognize the promoter sequence which is there on the DNA. So what are the promoter sequence? So we have seen the promoter region. So here this particular promoter sequence, so here the -10 and -35 regions are very, very important. So these 2 sequences are highly conserved in majority of the prokaryotic species. So here the sequences are highly conserved. That's why these 2 regions are called consensus sequences. And this particular -10 box, which is represented with this sequence like TA, TAAT, is called Tata box. So why it is called Tata box means, which is given, the name is given after the scientist David Pribnow, so that's why it is also called Pribnow box. And here the another consensus sequence is minus 35 box. So this particular promoter region is very, very essential. So while studying the sigma factor, we learned that, so this particular sigma factor is divided into 4 regions. So this the sigma factor alone is divided into 4 regions, like region 1, region 2, region 3 and region 4. So the region between the 1 and 2 will help in the loosening the non-specific interactions between the RNA polymerase and the DNA. And then the region 2.1 will help the sigma factor to recognize the core enzyme, and the region 2.4 will recognize this particular minus 10 box, Tata box or Pribnow box, whatever may be. So this particular minus 2.4 region will sigma 2.4 region of the sigma factor will helps in the recognition of the minus 10 box, and the region 4.2 region of the sigma factor will helps in the recognition of this particular minus 35 region. So this is how the sigma factor helps in the initiation process. Now we will see how the elongation process occurs. So in the previous diagram you have seen that this particular transcription bubble. So now this particular transcription bubble will move forward. So unwinding the DNA and rewinding the DNA behind the RNA polymerase. So this particular RNA polymerase will start moving in 5' to 3' direction on the antisense strand or template strand of the DNA molecule. So this particular unwinding and rewinding are continuous processes, and the size of the DNA is remains constant in the transcription bubble. So the important points to remember in the elongation step are, so the elongation occurs in 5' to 3' direction. So that is one important point to remember. And this transcription bubble while moving forward, so the amount of the DNA that is unwind will be rewind. So the amount of the DNA unwind and rewind will remain constant. So the number of nucleotides that will be added towards the 3' end of the growing RNA chain may be 40 to 50 nucleotides per second. So this is very speed process, and that occurs in the prokaryotes. So the number of nucleotides that added during the elongation process to the 3' hydroxyl group of the RNA may be 40 to 50 nucleotides per second, and like this particular RNA polymerase will move forward by unwinding the double strand DNA and rewinding the DNA behind the RNA polymerase, and the nascent RNA will extend from the RNA polymerase, so that it can be available for the ribosomes to start the process of translation. Why? Because in prokaryotes the transcription and translation occurs simultaneously. So the elongation process will start after the losing sigma factor. So this elongation process, so how far this particular elongation process will continue, that depends on the kind of the gene that is involved in the RNA synthesis. And finally, after synthesizing a proper RNA molecule, so this particular elongation step will continue until it reaches sequence called termination site. So that region, when that particular termination site is transcribed, the process of elongation will halt. So this is again explaining the process of transcription step by step. So here this is how the RNA polymerase will bind over the double-stranded DNA, just holding this particular RNA polymerase will tightly hold the DNA molecule, and this is called closed promoter complex. And after binding of the sigma factor and because of the conformational changes, this particular RNA polymerase will unwinds the DNA molecule, and this is called open promoter complex. And after formation of the open promoter complex, the one of the two strands will act as the template strand, and this particular RNA polymerase will synthesize, will add complementary nucleotides, and this occurs in 5' to 3' direction. So this is how the elongation will continue on the template strand, means which is coding for the RNA molecule. And then we will see how the termination occurs. So termination, so this particular elongation will continue until it reaches sequence, specific site, that is termination site. So what is termination site? So this termination is of two types, can occur in two ways in case of the prokaryotes. So one is row independent termination and row dependent termination. Row independent termination is also called intrinsic termination. Intrinsic means the term intrinsic represent inbuilt, because of the some inbuilt sequences this particular termination occurs. So that's why it is called row independent termination. So how the row independent termination occurs? So here an inverted palindrome sequence will be transcribed into the RNA molecule. So because of this particular inverted palindrome sequence, so that particular sequence results into the formation of a hairpin loop structure or a stem loop structure. And this particular hairpin loop structure, because of the formation of hairpin loop structure, the RNA the stability of the RNA polymerase is decreased, and that's why the RNA polymerase will dissociate from the DNA molecule. And not only the hairpin loop formation, because of the transcription of the adenines of DNA, the interaction between the adenine and uracil molecules is very weakest one, and finally the RNA polymerase will dissociate from the DNA molecule by halting the transcription process. So this is an inverted palindrome sequence. So this is how a stable hairpin loop structure or stem loop structure will be formed, and because of this particular stem loop structure formation, the RNA polymerase is unable to hold this particular growing RNA polymerase onto the DNA polymerase. So that's why this particular hairpin structure destabilizes the RNA polymerase, and ultimately the process of transcription will halt after synthesizing the hairpin loop structure formation. And another one is row dependent termination. Row dependent termination means here a specific protein, row factor or row protein is required for the termination process to occur. So this particular row factor is a hexameric ATPase. So means here the energy released during the ATP hydrolysis process is utilized by this particular proteins to dissociate RNA molecule from the DNA molecule. So this particular row proteins will bind onto the RNA molecule and will migrate to along with the RNA polymerase to some extent, and will halt the process of transcription. So this is how the row proteins will assist in the termination process. So once we will see, so during the termination process will occur when the RNA polymerase encounters a specific sequence called termination sites, and this is nothing but an inverted palindrome sequence, and after the transcription of this particular inverted palindrome sequence, a hairpin loop structure will be formed. So means intra-chain molecules will result into the hairpin loop structure, and because of this stem loop structure, the RNA polymerase will lose its stability and its ability to bind with the DNA, and finally it dissociate from the DNA molecule. And we will see, so this is how the Rho proteins will helps in the termination process. The Rho proteins will bind to the nascent RNA molecule and migrate along with the RNA polymerase, and the ATP hydrolysis process will finally makes the RNA polymerase to dissociate from the DNA molecule. So some important inhibitors like some antibiotics, these particular antibiotics will inhibit the transcription process, like rifampicin and rifamycin will inhibit the process of transcription process in case of the prokaryotes. This rifampicin is used for the tuberculosis treatment process. Not only these two antibiotics, actinomycin D also binds to the DNA molecule and prevent the RNA polymerase to bind to the DNA molecule and inhibiting the transcription process. So there are some important bits to study. So for your competitive exams, the about the RNA polymerase. So the question will come like this: if it is RNA polymerase 1, 2, 3, or RNA polymerase 1, which is involving in the transcription of the prokaryotes, and every question is important, like which enzyme involves in the DNA replication and what is the enzyme involves in the replication of prokaryotes. So this is how the first level of gene expression occurs. So that is transcription. So through the transcription, the first level of gene expression occurs, means the DNA transfers its information into the RNA molecule, and from the RNA the information is decoded by the ribosomes to produce the proteins. So this is so today we have learned about the first level of gene expression, that is transcription process. Thank you to the Commissionerate of Collegiate Education for giving me this opportunity. Thank you.